Hey everyone, welcome to The Explainer. Today, we're diving into something absolutely mind-blowing, the ancient origins of syncytins. Seriously, this is going to completely shatter and then rebuild how you view human biology. You know, when we think of viruses, we almost always think of disease, right? Pathogens, sickness, stuff we desperately need to fight off. But what if I told you that you wouldn't even be here today? Like you literally wouldn't exist as a human being without them?
45%. Let that number really sink in for a second. It's a massive, massive shock to the whole idea that our DNA is purely 100% human. Because it turns out nearly half of the human genome is actually made up of viral sequences. Almost half of what makes up your very own genetic code is just remnants of ancient viruses that infected our ancestors millions of years ago. It kind of completely redefines what it means to be human, doesn't it? So naturally, this leads us to a pretty fascinating question. How exactly did ancient viruses become a permanent part of us? Well, to solve this mystery, we have to turn to this incredible field of study called paleovirology. You can think of it sort of like archaeology, but instead of, digging in the dirt for dinosaur bones, scientists are actually searching right through the human genome for viral ghosts. These are basically fossilized genetic sequences that have been embedded in our DNA over millions of years of evolution. Section 1. The viral ghost inside us. Exaptation and domestication. For a really long time, scientists just dismissed these viral sequences as junk DNA, just leftover evolutionary baggage, doing absolutely nothing. But we're totally shifting that paradigm now to concepts called exaptation and domestication. Basically, our mammalian ancestors didn't just survive these ancient viral infections, they actually captured these viral genes and repurposed them for their own survival. They domesticated them, kind of like how early humans domesticated wolves into dogs. And that brings us to the absolute star of this explainer, syncytin. A syncytin is a domesticated viral gene derived from the envelope of an ancient retrovirus. In a normal infectious virus, this envelope protein is a weapon. It's what lets the virus latch onto and fuse with a host cell. But mammals actually co-opted this exact same mechanism to fulfill an essential physiological role. We literally took a weapon of infection and turned it into a tool for creating life. How wild is that? Section 2. Building the placenta, a biological marvel. To understand just how miraculous this is, we have to think about the massive biological challenge of live birth. For a mammal to actually carry a fetus, the mother's body needs to create this highly specialized temporary organ, the placenta. It has to exchange nutrients and oxygen without mixing blood, all while making sure her immune system doesn't reject the fetus. So how did mammals solve this incredible evolutionary hurdle? Well, they used a virus. When we zoom into the cellular level, we find a viral protein called syncytin 2 and its cellular receptor, MFSD2, driving cell fusion. In humans, we actually have two of these critical genes, syncytin 2, which is over 45 million years old, and syncytin 1, which is about 30 million years old. These ancient viral proteins drive single cells in the placenta, called cytotrophoblast, to fuse together into a giant, continuous layer called the syncytial trophoblast. This fused layer is the primary, absolutely essential barrier in the placenta between mother and fetus. Without this viral fusion protein, the placenta simply cannot form. But wait, syncytins didn't just give us the structural ability to fuse cells. They brought us another incredible gift. The crucial point here is that syncytins carry a functional immunosuppressive domain, or ISD. Think about it. When a mother is carrying a fetus, the fetus has DNA from the father, which essentially makes it foreign tissue. Under normal circumstances, the mother's immune system would violently attack and reject it. But that viral ISD suppresses the local immune response, protecting the growing baby. Now, really lean into the cognitive dissonance here for a second. This is the exact same mechanism that causes severe immunodeficiency in modern retroviruses like HIV. But in the placenta, we've basically harnessed that terrible immunosuppression for good to prevent maternal rejection. In fact, scientists believe this might have been the primordial function that caused our ancestors to capture these viruses in the very first place. Section 3. How viruses entered our DNA. The endogenization process. Now, you might be wondering, what are the actual mechanics here? How does a fleeting viral infection become a permanent, inherited part of a species across 550 million years of mammalian evolution? Well, it requires a very specific, mind-bending sequence of events at the molecular level called retrovirus endogenization. Let's break down how this works step by step. First, the virus can't just infect a random skin or lung cell. It absolutely has to infect the host's germline cells, so the sperm or the egg.
Second, the virus's DNA integrates itself directly into the host's genetic blueprint. Third, if that specific sperm or egg actually goes on to successfully create an organism, that viral DNA is vertically transmitted to every single cell of all future offspring. It becomes a permanent fixture. And finally, over millions of years, while most of the virus just mutates and rots away, the host domesticates the most useful viral genes, like that envelope gene we talked about, locking it in for its own evolutionary advantage.
Section 4. The evolutionary necessity. What happens without these genes? We can talk all day about how amazing this theory is, but in science, we actually need to prove it. We have to raise the stakes and ask, what happens if we remove these ancient viral genes from a living mammal today? Are they just kind of helpful, or are they truly essential? Well, scientists tested this by looking at mice, specifically mice missing the syncytin A gene and mice missing syncytin B. When scientists knock out that syncytin A gene, the cell layers in the placenta completely fail to fuse. The whole architecture of the early placenta just collapses, and the embryos sadly die at mid-gestation. Without this domesticated virus, the embryos literally cannot survive. It's wild, but this proves beyond a shadow of a doubt that this ancient viral gene is an absolute non-negotiable requirement for mammalian life. Section 5. Convergent evolution. Lightning strikes twice and again. If you thought that was incredible, prepare yourself for literally the biggest plot twist of this explainer. This miraculous capture of a viral gene to build a placenta, it didn't just happen once in some single ancient common mammalian ancestor. No way! It happened repeatedly, independently, across completely different animal families. When you map this all out, you see this incredible array of diversity. Carnivores, like dogs and cats, captured their own retrovirus over 80 million years ago. That's syncytin CAR1, which is the oldest one we know of. Primates, including us humans, captured syncytins 1 and 2 much later. Mice captured A and B. Rabbits grabbed one just 12 million years ago. Even ruminants, like cows and sheep, captured one specifically tailored to create their own unique type of localized cell fusion. Each distinct mammalian lineage was infected by completely different retroviruses at totally different times in history, and each one independently came to the exact same evolutionary solution, domesticating a virus to build a placenta. Evolution literally repeated itself over and over again.
And because our genomes are incredibly dynamic, we also see a concept called lost syncytins. It's basically a continuous cycle of retroviral capture, acceptation, and eventual evolutionary decay. For instance, an older viral gene called NV completely lost its fusion ability in humans, so it acts mostly as a genetic fossil for us today. But, and this is so cool, it is actually still active and fusing cells in certain monkeys. So it's this continuous, multi-million-year relay race, where ancient viruses eventually break down and newly captured viruses step up to take their place.
Section 6. The ultimate viral gift, redefining our relationship with viruses. As we bring this all together, I really hope your entire perspective on viruses has shifted. We've spent so much of our history viewing them purely as simple pathogens and enemies. But when we step back and look at the grand scope of our genome, they are so much more than that. They are literally the ultimate drivers of evolutionary innovation. And hey, syncytins aren't the only gifts we've received. Beyond the placenta, we have other amazing viral domestications. Take the FB1 gene, for example. It's actually a retroviral capsid that was domesticated 7 million years ago to act as an antiviral factor. We are literally using ancient virus parts to fight off modern viral infections. And then there's EBLIN1, this incredibly old 54-million-year-old non-retroviral gene that got inserted into primates. We are walking around heavily armored with the useful fragments of ancient pandemics.
There's this powerful quote straight from the source material that I just have to share with you. Viruses are not just harbingers of disease, but leads in the story of life on Earth. Just let the profound philosophical weight of that sink in for a second. The evolution of mammalian live birth, literally the very reason you and I are breathing, thinking, and checking out this explainer right now, simply wouldn't exist without viruses. Which leaves us with this final thrilling thought to ponder. If nearly half of our DNA is viral in origin, and we already know they give us the placenta and parts of our immune system, what other incredible hidden abilities have we inherited from ancient pandemics, just waiting to be discovered inside our own genetic code? It's like a giant puzzle. Keep exploring and absolutely never stop questioning what makes you, you.